Hello everyone, welcome to another MCAT ACE lecture. In today's lecture we are going to go over the digestive system, starting with description of the structure and functions of the digestive system, and then we will go over all the structures of the digestive system, starting with oral cavity all the way to the large intestine. We will also explain other organs, like for instance liver and pancreas, and their function in helping with the digestion and absorption of food. And then finally we will conclude our video by reviewing some of the major digestive enzymes as well as the gastrointestinal hormones, and then as always we will go over some practice questions, and that would be the end of the lecture after that. Alright, so here starting with the functions of the gastrointestinal tract. So basically the digestive system consists of the digestive tract as well as all the accessory organs, like for instance pancreas or gallbladder, that secrete fluids in the digestive tract. Now the major functions of the digestive tract include, for instance, ingestion, which refers to introduction of food into stomach. Then we have mastication, which is basically the same definition as chewing, and the purpose of chewing food is to break down food particles into smaller pieces, as a consequence of which there would be increased surface area to allow for better penetration of digestive enzymes. So basically this would increase surface area of food particles. Next we have propulsion, which is basically movement of food along the digestive tract that is being induced by peristaltic waves. So here movement of food along digestive tract by peristaltic waves. And these waves are consisting of contraction and relaxation that are induced by the circular smooth muscles in the digestive tract. So these are waves of contraction and relaxation that are induced by the circular smooth muscles. Next we have mixing of food with the digestive secretions, and then so here is mixing of food with digestive secretions, like for instance gastrin or hydrochloric acid is mixing with food. And then we have digestion, which is divided into two parts. One is mechanical digestion, which is being done by mastication as well as the segmental contractions, as a consequence of which it will break down food into smaller particles. And then we have chemical digestion that is being induced by the digestive enzymes. Then we have absorption, where molecules are being transported out of the digestive tract, and it could be done with either simple diffusion, facilitated diffusion, or active transport. And then finally we have elimination, where waste products are being excreted from the body as feces. Alright, so basically that is a summary of the functions of the digestive tract and different definitions that you should be familiar with. Now let's move on with different structures, starting with the oral cavity or mouth. So the first structure that we have in the oral cavity or mouth is teeth, which are used for mastication or chewing, as a consequence of which large food particles will break down into smaller ones, and so there would be increased surface area, and so therefore there would be better digestion. Then we have the tongue, the function of which is basically to move the food particles around and help with the chewing and swallowing, in addition to taste perception. So taste perception as well as for manipulating the movements of food particles. And then we also have the salivary glands, which include, for instance, the parotid, submandibular, which, as the name implies, is under your mandible, submandibular, and then sublingual, which is under your tongue. And so these are the salivary glands that are secreting saliva into the mouth, and these are an example of an exocrine glands. All right, so they produce saliva, and the purpose of the saliva is to moisten the food and also to lubricate the food for enhanced swallowing ability. It also contains lysozyme, so it has a lysozyme that will help kill the bacteria. And then in addition, the saliva contains the alpha amylase enzyme that will break down the carbohydrates. And so basically the breakdown of glucose molecules in the starch and other polysaccharides start in the mouth. So if you want an actual live example of alpha amylase breaking down these sugar molecules, try chewing on a small piece of bread and chew on it for about 1 minute. You will notice that after 30 seconds or so, now you're starting to notice a sweet taste to that piece of bread. And that is the reason that alpha amylase is breaking down those sugar molecules into glucose. All right. Now the next structure that we have is the pharynx, which allows movement of food into the esophagus. So from the oral cavity, with the help of the pharynx, which covers all this area right here, food is able to move to the esophagus. All right. Now, if you refer to different books, they would further break down pharynx into the upper area, which is close to the nose, and they refer to it as nasopharynx. The middle area that is close to the oral cavity, they refer to it as oropharynx. And then the lower area that is close to the larynx, they refer to it as laryngopharynx. But you don't really need to know that details. I'm just letting you know so that you would know what to expect in different books. But basically here we are having the pharynx that allows food to move from the oral cavity to the esophagus. Now, once food is moving into the esophagus, there is risk that some of this food enters the nasal cavity, and then some of it could also enter the larynx, and then and then through the trachea it goes to the lung, as a consequence of which it will cause choking. Now, in order to prevent all that, there are two structures. One is the soft palate that will block any food from pharynx to enter the nasal cavity. So again, the function of soft palate is to protect the nasal cavity and prevent any food from entering the pharynx into the nasal cavity. And then another important structure is epiglottis, which protects the larynx, and it prevents any food particle from entering into the larynx, and then from here on the trachea. So epiglottis is playing a very important role in preventing choking and having any food particle lodging into our lungs. Now moving on to the next structure. So we've discussed the oral cavity. From there on, we discussed the pharynx that allows food to enter the esophagus. Now this esophagus is basically consists of a muscular tube that allows the ingested food to pass from throat to the stomach. So from here on, food will enter the stomach. And now, because this is a muscular tube, it's not really easy for the food particles just to drop all the way down. And so in order for the food particles to move along the esophagus, they will have to go through the process of peristalsis, which basically consists of waves of relaxation of the circular muscles in front of the food bolus, followed by a wave of contraction behind the food particles. So here you can see as the food is going through, the front area is relaxing to let the food go through, but then the area behind the food bowels is undergoing contraction. So these waves of contraction and relaxation are referred to as peristalsis, and these are being induced by the circular muscles. Now, what happens when you have something toxic in your stomach or your stomach is irritated? You will vomit. And vomiting is actually reverse of the peristalsis process. And then some other books may refer to it as emesis, and these are all the same definitions, vomiting or emesis. And then one other point that I would like to mention is that at the end of the esophagus, there is a sphincter that is called the lower esophageal sphincter. And so the function of the sphincter is to allow the food into the stomach, and then it closes to prevent the food reflux back into the esophagus. Because think of it, the stomach is full of the acidic fluids, and we don't want any acidic fluids to regurgitate back into the esophagus, because it would cause damage to the esophagus, and if it continues to do that over long term, it can lead to esophageal adenocarcinoma, which is a form of cancer. So therefore, the function of the lower esophageal sphincter is to prevent any regurgitation of food back from the stomach into the esophagus. Now, patients that have gastroesophageal reflux disease, these patients are actually suffering from regurgitation of fluid from stomach into the esophagus, as a consequence of which they are at increased risk of developing esophageal adenocarcinoma, which is from a long-term exposure of the esophagus to the stomach acid. Alright, so from esophagus, food has entered the stomach, and stomach is basically a thick-walled organ that lies in between the esophagus and duodenum. So the next structure from the stomach that we will encounter is the duodenum. So up to here is the esophagus, and then from here on, the duodenum starts, and here we have the stomach. The stomach itself is divided into different parts. The fundus is the upper portion, the body, antrum, and then here finally we have the pylorus before food enters the duodenum. Now, two other structures that I would like to mention here is one is the pyloric sphincter, which controls flow of food from stomach into the duodenum. So the pyloric sphincter, similar to the lower esophageal sphincter that was controlling entering of food into the stomach, here the pyloric sphincter controls entry of food into the duodenum. And then the wall of the stomach is also containing rugae, which are basically gastric folds. And these gastric folds allow the stomach to expand during food intake. And so that's the reason that the stomach can hold a large amount of food, because it contains so many gastric folds that can expand once we start to eat food. Now, a combination of acid plus enzymes plus hormones that are released by the stomach, all these combinations are referred to as gastric juice. And there are four major cell types in the stomach that helps with the production of the gastric juice. So now let's go over the functions of different cell types in the stomach. So the first cell type is the surface mucous cells, the function of which is to produce the alkaline mucous surface. And this alkaline mucous layer is actually protecting stomach against HCl and pepsin. Because think of it, stomach is basically a muscular structure. If we don't have any layer to protect it, the hydrochloric acid as well as the pepsin, which is the enzyme that breaks down proteins, they will eat through the stomach. So we need a layer to protect stomach from being digested, and that is being done with the help of the surface mucous cells that will producing the alkaline mucus layer. Now there are conditions that can destroy this alkaline mucus surface, and these include, for instance, aspirin or Advil, the other name for which is ibuprofen. These can destroy the surface mucus layer. And an infection, for instance, with Helicobacter pylori can destroy the mucus barrier, as well, as a consequence of which these patients are at increased risk of stomach ulcer. Indeed, if you go over some of the adverse effects of taking aspirin or ibuprofen, you will see that stomach ulcer is way high on the list. Now the next cells that we have are parietal cells, the function of which is to produce the hydrochloric acid. And so this hydrochloric acid will decrease stomach pH, and once this stomach pH is activated, it will help activate pepsin, which is produced by the chief cells. Now the other function of the parietal cells is to produce intrinsic factor, which binds to vitamin B12 and help its absorption in the ileum. So let's go over the functions of the parietal cell again. It produces HCl to lower the pH of the stomach, and it produces the intrinsic factor that helps with the absorption of B12. Now chief cells, on the other hand, are producing pepsinogen. So let me erase all that. So chief cells are producing pepsinogen, which is basically a zymogen. And can you tell me, what does zymogen mean? Basically, zymogen means an inactive precursor. So this is an inactive enzyme, and so that's why we are referring to it as a zymogen. So in order for it to become active, it needs to be converted into pepsin. And this pepsin conversion is being done with the help of the low pH that induces conformational changes that make the pepsinogen undergo cleavage to turn into its active form called pepsin. Now pepsin is a proteolytic enzyme that will break down proteins into smaller peptide molecules. And then the final type of cell that we have here are G cells, which are required for the production of gastrin hormone. So G cells produce gastrin hormone, and the function of gastrin hormone is to increase production of hydrochloric acid and also induce gastric emptying by increasing the stomach motility and also by relaxing the pyloric sphincter. So recall here we had the pyloric sphincter. G cells are releasing gastrin in order to relax the pyloric sphincter and allow food to enter from stomach into the duodenum. Now the next point that I would like to mention is that depending on their functions, the organelles that you find in different type of cells could be different. For instance, parietal cells, they need to supply energy in order to pump hydrogen ions into the stomach. So they require a lot of energy in order to pump hydrochloric acid into the stomach. As a consequence of which, these cells are rich in mitochondria to supply the energy needed. And then the other cells, like for instance chief cells or G cells or the mucus producing cells, these now are requiring a lot of rough endoplasmic reticulum as well as Golgi bodies in order to produce different proteins. And so therefore, depending on their needs, the type of organelles that you find in different cells, the proportion of it could be different. So remember, parietal cells have more mitochondria because they need to produce more energy. Moving on to the next section, which is the liver gallbladder, and bile. And these are basically the accessory organs that we're talking about that will help with the digestion of food. So starting with the liver, basically liver is the largest organ in the body that is located on the right side of your stomach, and it's actually inferior to your diaphragm. It's required for many different functions, and I'm just gonna quickly overview those for you. Like for instance, it's required for metabolism of lipids, carbohydrates, and proteins. It can be also used for storage of fat and triglycerides. So it's required for metabolism of lipids, carbohydrates, and proteins. It can also be used for storage of fat. They're also required for storage of glucose as glycogen. They can help with the detoxification of different drugs. So they can be used for detoxification of different drugs or breakdown of different hormones. They are producing the clotting factors. So next we have the clotting factors. They produce albumin. And then they help convert ammonia into urea, which is less toxic than ammonia and can now get excreted in the kidneys. And then the final important function of liver that is very related to our current lecture is production of bile that helps with the emulsification of fats. And so bile that is produced here is directly draining into the duodenum. So bile can go directly to duodenum, and some of them is being stored in the gallbladder. So the function of gallbladder is storage of bile. Now speaking of bile, let's go over the function of bile next. One question that so many students ask is that what's the difference between bile acids and bile salts? We keep hearing like secretion of bile acid versus bile salts, and there's sometimes confusion. So let's go over the difference between those. Cholesterol that enters the liver is being converted into bile acids. And then bile acid, once they are conjugated with amino acids, this combination is now called a bile salt. And then it's the function of bile salt to help with the emulsification of fat. And then how bile salt actually do that is that they are composed of a hydrophobic portion, here shown in yellow, and hydrophilic portion that is shown in green. So once bile salts are introduced into a fat globule, they would bind to the fat particles and form emulsified micelles. And so these fat droplets can no longer aggregate back. And so by doing this, there would be increased surface area of fat particles in order to be better digested by the lipase enzyme. And then the final point that I would like to mention in this slide here is that if you've noticed, the pancreatic enzymes as well as the liver products and the gallbladder bile, all of them are being emptied into the duodenum at the same site. So here we have the liver via the common hepatic duct and gallbladder via the cystic duct. They join into common bile duct. So here we have the common hepatic duct and then the cystic duct. They join together to form the common bile duct. And then common bile duct and pancreatic duct, they will join together, and here at the hepatopancreatic ampulla, they would empty their contents into the duodenum. All right, so if you need to pause the video to review these structures, you can do that. Next, we have pancreas, which is located on the left side of the body. So we just discussed how liver is on the right side. So here we have liver and gallbladder on the right side of the body. Pancreas is, on the other hand, on the left side of the body. So let's go back. All right, so pancreas is located on the left side, and it contains both the exocrine cells, which are required for digestion of nutrients, and then we have endocrine cells that are required for production of hormones. And we are not going to go into too much details regarding endocrine cells because we are having an endocrinology video that we are discussing in further details, different hormones that are being produced by the pancreas, but we will briefly touch upon these topics right here. All right, so starting with the exocrine, these cells basically contain inactive enzymatic precursors. And can you tell me what was the name for inactive enzymatic precursors? They were called zymogens. So basically, pancreas contains zymogens. And if you think of it, it all makes sense. You don't want all these digestive enzymes to get activated in the pancreas because they will eat through the pancreas. So they are being produced as inactive forms. And these enzymes include trypsinogen, chymotrypsinogen, amylase, the function of which was to break down carbohydrates. We just discussed the salivary glands produce amylase, so does the pancreas. 4, we have the lipase, and then we have deoxyribonuclease, which will break down DNA, and we also have ribonuclease, which breaks down RNA. And these are all being secreted into ducts that are arranged in clusters called acini. So here we have the acini. All these enzymes that we just discussed are being produced right here. All right, now moving back to these enzymes. So these were again the inactive zymogens. Now trypsin and chymotrypsin. So these two will become active with help of the enterokinase enzyme that is present in the intestinal mucosa. So once these zymogens are released into the intestine, the enterokinase enzyme that is present in the intestinal mucosa will activate these two enzymes. And now after trypsin has become activated, it will activate all the rest of the enzymes right here. So now we are having all enzymes activated. And so pancreatic amylase will now continue to digest the polysaccharides. Pancreatic lipase will break down lipids into smaller molecules, like for instance glycerides, fatty acids, and cholesterol. And then deoxyribonuclease and ribonuclease, these will help digest DNA and RNA. And then as for trypsin and chymotrypsin, these are proteolytic enzymes that will break down protein. Now, in addition to all these secretory enzymes, exocrine cells also produce bicarbonate. And the reason bicarbonate is being produced is that it helps neutralize acidic content of the stomach that enters the duodenum from the stomach. So if you think of it, here we have duodenum, and all the content of the stomach is entering the duodenum, which is pretty acidic. So by having some bicarbonate enter right here into the duodenum, it will neutralize the acidic content. And so that was all the functions of the pancreas, pancreatic exocrine cells. Next, we have the endocrine cells. Again, I'm not going to go into details here, just briefly touching upon different hormones. So we have 3 types of pancreatic endocrine cells: beta cells that produce insulin, alpha cells that produce glucagon, and then delta cells that produce somatostatin. And the way I memorize it is I call it betunsulin. And so this is my own memory aid. I remember that beta cells produce insulin. All right, now let's go over the function of different hormones right here. So first we have insulin. And the function of insulin is to enhance absorption of glucose into muscle and adipose cells, as a consequence of which there would be decreased blood sugar. So in diabetic patients, if there is increased resistance to insulin, and that would be type 2 diabetes, or if there is decreased secretion of insulin, like from having autoimmune destruction of the pancreatic beta cells, that would be type 1 diabetes, where there would be no production of insulin. In these patients, since there is no insulin, their blood sugar will keep rising. So that's the function of insulin. Next we have glucagon. The function of which is reverse of insulin, so it increases blood sugar. And then finally, we have somatostatin, which inhibits the growth hormone. Now, I would like to mention here that you know how all these acini are releasing their contents into the pancreatic duct. The endocrine cells don't do that. All these islets are releasing their contents into the bloodstream. And that's one important definition of hormone, in that they're distributing to different parts of the body via the bloodstream. All right, next we have the small intestine, which is basically consists of 3 parts. It consists of the duodenum, which is the shortest part of the small intestine, jejunum, and then ileum. And then we discussed how all the contents of the pancreas as well as the bile from gallbladder are emptying into the duodenum. So basically, duodenum is the site of digestion of food particle, while ileum and jejunum, these are basically the site for absorption of food particles. Now, in order for the small intestine to absorb food particles, it needs high surface area. And for that, they have actually finger-like projections called villi. So here we have a cross-section of the small intestine, and you can see that here we have a finger-like projections from the intestine that are called villi. And the function of villi is basically to increase the surface area. Now, inside all these villi, there is a capillary network that will help absorb most of the nutrients, but then there are some large macromolecules and fat molecules that cannot get absorbed into the capillary network, and those are being absorbed through the lymphatic vessels shown right here, the other name for which is lacteal. Now, this lymphatic vessel doesn't really have a basement membrane, unlike the capillary network that has basement membrane. Lacteal doesn't have a basement membrane, and so therefore it can absorb larger molecules like macromolecules and fat molecules. Now, I'd like to mention that the villi themselves also have cytoplasmic extensions on the surface of their cells that are called microvilli. So while we are having these finger-like projections, the fingers themselves have cytoplasmic projections that are called microvilli that would further increase the surface area of small intestine for enhanced absorption. Now, the digestive enzymes that you find in the small intestine, they are actually located on the microvilli that are shown right here. So all the digestive enzymes are bound to the microvilli, and they're called brush border enzymes. So the small intestine enzymes that are bound, all of them, to the microvilli, these are referred to as brush border enzymes. We also have crypts here, which are located at the base of the villi, and they are referred to as crypts of Lieberkühn. And these crypts contains the glands that produce the intestinal juice as well as lysozyme, the function of which is to kill bacteria. So now that we've discussed the structure of the small intestine, let's go over different segments of the small intestine. So we just discussed that duodenum is the shortest part of the small intestine and is the site where all the gastric content as well as bile and pancreatic juice, which includes the enzyme and bicarbonate, are being released into this section. And so therefore duodenum is the main site for digestion of food. Iron is also being absorbed in this section. But then one other important function of duodenum is that because all the acidic chyme from the stomach has entered the duodenum, even though the pancreas releases bicarbonate into duodenum, there is still increased risk of damage to the duodenum. And so in order to prevent self-digestion from acidic chyme of the stomach, duodenum has three main functions. Number one is that it produces a mucus layer by goblet cells to protect from digestion. Two, it uses up the bicarbonate from pancreas to neutralize the acidic chyme. And then finally, it has Brunner's glands that are secreting an alkaline mucus in the duodenal submucosa to protect against stomach acidic chyme. All right, so via production of mucus layers by goblet cells, as well as production of the alkaline mucus by Brunner glands, the duodenum is able to protect itself from acidic chyme. And then finally, the bicarbonate that is released from pancreas also help neutralize the acidic chyme in the duodenum. So that's the function of duodenum. Now let's move on to the next section, which is jejunum. So we already discussed duodenum up to here. That's the shortest part of the small intestine. Next we have the jejunum, which is shown in light blue. And basically this is the midsection of small intestine that connects the duodenum to the distal part that is called ileum. And then the function of the jejunum is actually to help with absorption of digestion products of duodenum, like for instance carbohydrates, amino acids, and then fatty acids. And then the final section is the ileum of the small intestine, which is required for the absorption of vitamin B12. So let me pick a different color to distinguish these. So ileum is required for absorption of vitamin B12. It also helps with absorption of fat-soluble vitamins. And can you tell me the name of the fat-soluble vitamins? Vitamin D, vitamin E, vitamin A, and vitamin K. These are all examples of fat-soluble vitamins. So these are also being absorbed in the ileum. And then finally, bile salts are being absorbed also inside the ileum. Now I'd like to remind you that here for the absorption of vitamin B12, we need the intrinsic factor, which were produced by the parietal cells. So here the intrinsic factor that is bound to vitamin B12 helps with the absorption of this vitamin. So let me show it to you right here. So here we have parietal cells, and I'm sorry I erased it, but here we had the intrinsic factor that were produced by the parietal cells to help bind to vitamin B12 and helps with the absorption in the ileum. All right, moving back to the small intestine. So that's it. The vitamin B12 is being absorbed with the intrinsic factor. Now the next section that we have is the large intestine or colon. And this section starts from the end of the ileum. So once the ileum ends here, now here starts the large intestine that goes all the way through here to the anus. Now different sections of the large intestine include cecum, is the very first part of the large intestine. Then we have the colon. Then we have the rectum, and then finally we have the anal canal. And those are being shown right here in this image. So the first part is called the cecum. Then we have the ascending colon, the transverse colon, the descending colon. Then we have the sigmoid colon, and then finally the rectum and the anus. And then the major events that leads to the formation of feces in the colon include, number one, absorption of water and salts. And so I recall we were asking in medical school from our instructors that isn't it the job of the kidneys to absorb water and salts? And we were always told to think of colon as a large kidney. One of the major functions of colon is similar to kidney, to absorb water and salts. All right, so keep that in mind. Large intestine is absorbing a large amount of water and salt. Indeed, if there is problem with the absorption of water and salt, that's the time that you can have diarrhea. Now the next function is secretion of mucus to protect surface of the intestine. And then finally, there is metabolic activity by microorganisms. So there are so many bacteria in the colon that are actually helping with breakdown of food particles as well as production of vitamin K or vitamin B7, for instance. So these are all good bacteria that we need them. Furthermore, these bacteria are preventing growth of other harmful bacteria. So we don't want to take too much antibiotic. That's why they tell you do not take too much antibiotic, because you will destroy flora of your colon, as a consequence of which now harmful bacteria like Clostridium difficile can start growing and cause diarrhea. So besides the metabolic activity, these microorganisms are also preventing growth of harmful bacteria. Now next here we have the summary of all the different structures of the digestive system, which includes the digestive tract as well as all the accessory glands and organs. All right, so if you need more time to pause this section and review all the different structures, by all means, but otherwise we have discussed every one of these structures through our video. Now here let's have a quick summary of enzymes that are being released by different glands or organs right here in the digestive tract. So first structure that we talked about was the salivary glands that are producing amylase, and their function was to break down carbohydrates into smaller molecules, like for instance glucose. We also discussed that pancreas is also producing amylase. And then small intestine can also produce enzymes that can help break down the carbohydrates. Next, we have the gastric glands in the stomach that are producing pepsin, the function of which is a proteolytic enzyme to break down proteins into smaller peptides. We also discussed that pancreas also produces trypsin and chymotrypsin that helps with the breakdown of proteins. And then we also have carboxypeptidase produced by pancreas. We didn't discuss it earlier. This enzyme also has a similar function to trypsin and chymotrypsin. And then small intestine also has glands that are producing the peptidase enzyme. And then finally, we have the lipase that is being produced both by the salivary gland and pancreas, but mainly by the pancreas it's being produced. And then gallbladder, bile micelles, these are not really an enzyme, but I listed them here because they would help with the emulsification of the lipids in order to increase the surface area. And now the lipase can act on all these emulsified micelles to help with the breakdown of fat particles into glycerol and fatty acid. So that was a quick summary of all the enzymes that are produced in the digestive tract. Now let's go over the hormones that are produced in the digestive tract. So here I'm not discussing the hormones that are produced in the pancreas because that's not related to digestion. Here I want to go over the hormones that help with the digestion process. So we discussed already the gastrin that is being produced by G cells, and a secretion of gastrin is being initiated by stomach distension, meaning that food has entered, or by digested proteins, or stimulation by the vagus nerve, which is a parasympathetic nervous system. And upon release of G cells, there is increased production of hydrochloric acid, and we also discussed that there would be increased stomach motility as well as relaxation of pyloric sphincter in order to allow the food to enter from stomach into duodenum. Now, once food enters the duodenum needs time to digest all those. So there will be hormones that are secreted that will decrease the gastric motility. So duodenum is like, hold on a second, I don't want any more food. Give me time to digest all these. So all the hormones that are being secreted here, including secretin, cholecystokinin, or gastric inhibitory peptides, they would decrease gastric motility. At the same time, these hormones increase production of, for instance, like bile and pancreatic bicarbonate to help with the digestion of all the products that are now inside the duodenum. And then they would inhibit production of further gastric secretion because they are holding off. They're putting everything on hold in the stomach until they're done finishing their own job, which is digestion of the acidic chyme that has entered the duodenum. All right, so basically that concludes the discussion of our digestive tract. Now, there are some questions that I didn't address in our video, and I would like to hear addressed as short questions right here. And the first question is, what are the 4 layers of the digestive tract? So basically, the digestive tract consists of 4 layers that is present all the way from esophagus to the anus. And from inside to outside, these layers include the mucosa, submucosa, muscularis layer, and serosa or adventitia. So let's go over the function of every one of these layers. So first we have the mucosa, which is the innermost layer. It basically consists of the squamous and columnar cells, and the function of this layer is to produce a gastric mucus to lubricate food, and then at the same time it will produce a protective layer over the epithelium to prevent digestion of the stomach by pepsin. Furthermore, this mucosa can also secrete bicarbonate, which helps neutralize pH of the acidic contents that are being released into the intestine. Next, we have submucosa, which is the layer underneath the mucosa, and this layer is actually containing nerves, blood vessels, as well as glands. And then the nerves that are being found here in this layer, like an example is parasympathetic ganglions known as Meissner plexus. Next, we have the muscularis layer, which is mostly consisting an inner layer of circular smooth muscles and an outer layer of longitudinal smooth muscles. So this was the inner layer, and then the outer layer is longitudinal smooth muscle. And this muscular layer contains a nerve plexus known as myenteric plexus. And so a combination of activity of myenteric plexus and Meissner plexus, these two control movements and secretions within the digestive tract. So basically there is a local control of movements or secretions in the digestive tract with the help of all these enteric plexus, more specifically the Meissner plexus and the myenteric plexus. And then finally we have the serosa or adventitia, which consists a layer of connective tissue. And basically here this table summarizes all the findings. We have the mucosa, which is made of the epithelium, lamina propria, as well as the muscularis mucosa layer. Then we have the submucosa that contains the Meissner plexus. Muscularis layer contains an inner circular and outer longitudinal, and then the other name for myenteric plexus is Auerbach's plexus. And then finally we have serosa or adventitia, which is the outermost layer that contains the connective tissue. Alright, the next question is, what is the functions of lacteal? And we discussed this in this video. Basically there are macromolecules and fats that cannot get absorbed by the capillary plexus in the intestine, and so it's the function of these lymphatic vessels known as lacteal to absorb those macromolecules. The next question is, what happens if zymogen becomes activated in the pancreas? Well, if they become activated too early, then they would cause autodigestion of the pancreas, as a consequence of which there would be inflammation of the pancreas known as pancreatitis. And that's actually a phenomenon that is commonly seen in alcoholism. All right. Next, describe the function of different lipoproteins in the body. So I'm sure that all of you have heard of low density lipoprotein, or LDL, or HDL, which stands for high density lipoprotein, or very low density lipoprotein. But what is the function of every one of these lipoproteins? So let me ask you a question. First thing first, what does even lipoprotein mean? All right, so given that these lipids are water insoluble, they cannot really get transported in the blood unless they're being combined with some sort of protein that are water soluble. So once a protein binds to these lipid molecule, the combination is called a lipoprotein. And these lipoproteins are listed right here. So let me go over the function of every one of them. So first we have the chylomicron. So once food is inside the intestine, the smooth endoplasmic reticulum will attach proteins to fat particles, like for instance triglycerides, phospholipids, and cholesterol, and that combination is to form a chylomicron. Now chylomicrons will now leave the intestinal cells with the help of what structures? Lacteals. And so they would enter the bloodstream through these lymphatic vessels, and they would go through until they reach the fat and muscle cells, inside which, with the help of the lipoprotein lipase, they would release the free fatty acids into the adipose or muscle tissues. And then the leftover lipids are being transported into the liver. Now all the triglyceride and cholesterol that is being stored in the liver, they would get transported to all the peripheral tissues with the help of the very low density lipoprotein or low density lipoprotein. And then finally we have high density lipoprotein that transports all the fat from peripheral tissues, like here you can see from adipose and muscles. With the help of the LSAT protein, they would transport everything from the periphery to the liver. And so that's the reason that it's always an indication of good health when you have high density lipoprotein. And the reason for that is that all the fat that could cause trouble outside, like for instance if you have too much cholesterol outside in your blood, it can cause blockage of the vessels. So HDL is taking away all these excess fat and cholesterol back to the liver, inside which it can be broken down into bile and get excreted. So that's why we always want to have high HDL, which is an indication of good health. On the other hand, if you have high VLDL or high LDL, that's a bad sign, because it means that your liver is sending too much of these fat molecules to the peripheral tissues, which can cause, again, blockage of all the different vessels that can lead to heart attack or stroke or other problems. All right, the next question is, you might have heard the term jaundice. What is jaundice? So it's basically the yellow discoloration of the skin, so yellow skin color, that is from buildup of too much bilirubin, and that is called jaundice. So you know how bilirubin is normally getting excreted by liver into the intestine? Now if there is some problem where there is obstruction of the cystic duct, whether there is a gallstone or cancer of the pancreatic head, the bilirubin, which is actually the byproduct of the hemoglobin breakdown from blood, cannot get excreted, as a consequence of which there would be yellow discoloration of the skin, known as jaundice. Next question, what is the composition of bile, which is secreted by what organ? Gallbladder. Alright, so basically bile is mostly composed of water, so about 97% of bile is water. Then a small proportion of it is made of bile salts, which are required for emulsification of fats, and we just discussed how inside the liver cholesterol is being converted into bile acids that are being conjugated with amino acids in order to form bile cells, and these bile cells can now emulsify fat particles. Now bile is also composed of a small amount of bilirubin, which is a byproduct of hemoglobin breakdown, and then it also contains like other fat molecules, like for instance cholesterol or fatty acids. The next question is, we keep talking about villi in the intestine, but what's the difference between cilia and villi? So basically cilia are hair-like projections that you see on the surface of the respiratory epithelia, like for instance in your lungs, that help push all the particles outside upward towards the oropharynx. Villi, on the other hand, are found in the small intestine. So villi are found in the small intestine, while these cilia that are hair-like projections are found in the respiratory tract. Now, what are the roles of leptin and ghrelin in feeding? The reason I included this question here is that these are hot topics that could show up as a passage on your MCAT exam. So I thought it would be good for you to know the actual function of these proteins. So these hormones are playing a major role in energy balance and feeding behavior. So let me pick a different color here for you. So there we go. So leptin, it inhibits food intake, and so therefore increased secretion of leptin helps with weight loss, while ghrelin, on the other hand, promotes meal initiation, which is to start eating food, and so therefore it can cause obesity. And then in obese patients, you may notice that they are either leptin resistant or they have too much ghrelin secretion. All right, next question. What is the function of appendix? So basically appendix, its exact function is not known, but it's thought that it may help replenish the normal bacterial flora in the colon. So once you're having diarrhea and most of the bacterial flora in the colon are lost, appendix helps replenish the normal bacterial flora. And then the final question is that why is it that patients with liver disease like hepatitis develop ascites? And then I included the definition of ascites. It's basically buildup of fluids in the peritoneal or stomach cavity. All right, so if you may recall, one of the functions of liver is to produce albumin, which is a protein that is found inside blood. Now here, if you're having a blood vessel, there is a hydrostatic pressure that is trying to push the fluids out, while there is an oncotic pressure that is trying to pull the fluid inside. So hydrostatic pressure is developing from blood pressure, while oncotic pressure is an osmotic pressure that is being produced from albumin and other proteins in the blood. Now when you're having liver disease, there would be decreased production of albumin. So basically the osmolarity in terms of the protein content drops, and so therefore now there is decreased oncotic pressure, as a consequence of which more fluid will leak out of vessels. So that's why the patients that have liver disease, they will develop ascites, which is fluid buildup in their peritoneal cavity. All right, we are done with all these supplementary questions. Now let's do some practice questions. All right, so number one, textbooks say that the surface area of human gut is about the size of a tennis court. Absence of which of the following structures make this statement false? Next, we have the stomach is primarily involved in the digestion of which of the following nutrients? The next question, a patient suffers from lack of hydrochloric acid production in the stomach from defects of certain stomach cells. What other findings are likely to be observed in this patient? All right, so think of it, which stomach cells are producing a hydrochloric acid and what other products are being produced by that cell. Once you answer that, you should be able to answer this question. The next question is, which of the following products are being excreted from pancreatic duct? Which of the following is a true statement regarding pepsinogen? And then finally, which of the following products are produced by the pancreas? Right, so again, if you need to pause the video, by all means, go ahead. Otherwise, we are ready to go with that. All right, so basically here it's asking you what structure is helping to increase the surface area of the gastrointestinal tract. And it's actually the villi that are increasing the surface area. We know that pyloric sphincter is controlling movement of food from stomach into duodenum. Bile cells help with emulsification of fats, and rugae are helping with extension, like increasing the size of the stomach. But this extension is not nearly as much extension as the surface area induced by villi. So villi are responsible for increasing the surface area of the gastrointestinal tract. All right, next question is, the stomach is primarily involved in digestion of which of the following nutrients? So if you recall, we had mucosal cells that are producing mucus, we had G cells that are producing gastrin, and then we had parietal cells that are producing HCl and intrinsic factor for B12 absorption. And then the other important cell was the chief cells that are producing pepsinogen that via the help of HCl is converting into pepsin. And what was the function of pepsin? It's a proteolytic enzyme that will break down proteins. So stomach is primarily involved in digestion of proteins. Next question. A patient suffers from lack of hydrochloric acid production in their stomach from defects of certain stomach cells. So which cells are defective in these patients? Can refer back to this question. If they're not producing HCl, therefore it's their parietal cells that are having problem. And so if parietal cells are having problem, what other protein is not being produced? Intrinsic factor, which is required for B12 absorption. So what other findings are likely to be observed in this patient? The answer is deficiency of vitamin B12. Now, other answer choices are development of early stomach ulcers. No, because the mucosa is not being affected. Lack of trypsinogen activation. Well, trypsinogen is being activated by enterokinase that is found in the intestinal epithelial cell. So again, has nothing to do with defect in the parietal cells. And then finally, decreased production of pepsinogen. Pepsinogen is being produced by chief cells. If it was saying decreased activation of pepsinogen, it could have been a correct answer. But here it says decreased production of pepsinogen. No, pepsinogen is being produced by chief cells. All right, so choice B is the correct answer. Next question. Which of the following products are excreted through the pancreatic ducts? All right, so enterokinase, we just discussed, this is the enzyme that is found on the intestinal epithelium to help activate the trypsinogen and chymotrypsinogen. So this is not being secreted by the pancreas. Insulin is being secreted by the pancreas, but it's being released directly into the blood, not the pancreatic duct. So this is an incorrect answer too. Trypsinogen, yes, that is being released into the pancreatic duct. Glucagon, no, that is being released into the blood. So the answer is choice A. Trypsinogen is the only one that is being released into the pancreatic duct. Then the next two question is that which of the following is a true statement regarding pepsinogen? It requires an acidic environment to change conformation necessary for its activation. That is correct. It requires enterokinase for its activation. No, enterokinase is required for activation of trypsinogen and chymotrypsinogen. All right. Now it becomes activated in the alkaline environment. No, that's not correct. And then produced and secreted by the parietal cells. Again, that's not correct. Which cells produce pepsinogen? Correct. Chief cells are producing pepsinogen. Then the next question is, which of the following products are being produced by the pancreas? Hydrochloric acid, no. And here, let me pick a different color. Hydrochloric acid, no. That is being produced by the parietal cells. Emulsifier, again, no. That is being produced by the liver and released by the gallbladder. So the bile here we are talking about. Proteolytic enzymes, yes, like chymotrypsin and trypsin. These are all examples of proteolytic enzyme. So the correct answer is choice C. Now moving on to the last sets of questions. Again, if you need more time, you can go ahead and pause the video. Which of the following is not a function of the liver? So we discussed extensively different functions of the liver. Which of the following structures carry nutrient-rich, oxygen-poor blood? All right, this we didn't really cover in our video, so there would be a good chance to discuss the difference between all the vessels that are listed here. Moving on, which of the following findings are likely to develop in a patient that is on chronic antibiotic therapy? We already touched upon this topic. And then the last two questions is, which of the following hormones induces contraction of gallbladder? And then which of the following lipoproteins help cholesterol move from periphery to the liver? All right, so if you need more time, by all means, you can go ahead and pause the video. Otherwise, we are ready to start. So which of the following is not a function of liver? It's required for production of erythrocytes during fetal life. That is correct. So we do have a cardiovascular video inside which we have discussed that yolk sac, spleen, and liver are required mainly for production of blood during the fetal life, but later towards the end of fetal life and during adulthood, it's the bone marrow that takes over. So that is correct, even though we didn't discuss it in this video. It's required for detoxification of drugs. We discussed it in this video. It's required for storage of glucose as starch. No, it should be storage of glucose as glycogen. All right, and then it's required for destruction of damaged red blood cells. That is correct. So spleen is the major organ that is required for destruction of red blood cells, but bone marrow and liver can also destroy some of the damaged red blood cells. So the best answer here would be choice C. Moving on to the next question. So here I have provided you with an image that summarizes actually the function of every one of these vessels. That would be a good summary to go over. So there we go. All right, so once blood from aorta reaches the intestine, it's oxygen rich, but inside the small intestine it will lose the oxygen, but instead it picks up all the nutrients. So this blood is now nutrient rich but oxygen poor, and it's with the help of the hepatic portal vein that this blood enters the liver. So the answer here is, which of the following is nutrient rich, oxygen poor? And the answer is hepatic portal vein. Then we also have hepatic artery, which supplies the oxygen to the liver, and this vessel is directly arrived from the blood that comes from the heart. It doesn't go through the intestine. And then finally we have another duct called hepatic duct that will help release the bile into intestine, and these structures are usually arranged in a close proximity, so you can find all three of them near each other, and that's the reason that they are referred to as portal triad, which is composed of the hepatic duct, hepatic portal vein, as well as hepatic artery. And then the blood from the liver will enter the hepatic vein, and from here on it will go to inferior vena cava and then the right atrium. Which of the following findings are likely to develop in a patient that is on chronic antibiotics? So without looking at the answers, what does chronic antibiotic do to the gastrointestinal tract? It will destroy most of the bacterial flora that is required for production of vitamin K, for instance. So here there would be reduced production of vitamin K. That is correct. There would be reduced absorption of vitamin K. No, absorption of vitamin K, which is a fat soluble, is being done in the ileum. It has nothing to be done with the antibiotic therapy. It increases risk of infectious diarrhea. We discussed this. Yes, that's correct. Because once the normal bacterial flora are dead, now the opportunistic bacteria can take over and cause infection and diarrhea. It decreases risk of infectious diarrhea. Nope, that's incorrect. So 1 and 3 are correct here, and it would be choice C. Which of the following hormones induces secretion of gallbladder? And the answer is cholecystokinin. So if you go back to the table that I've provided for you here, you can see that the function of cholecystokinin was to induce contraction of gallbladder in order to secrete the bile salts. And then gastric inhibitory peptide inhibits secretion of gastrin. And then secretin inhibits the gastric motility and decreases release of the content inside the duodenum. And then finally we have the gastrin that induces secretion of hydrochloric acid. Now the final question that we have here is, which of the following lipoproteins can help carry cholesterol from periphery to the liver? So if you recall, we discussed we have chylomicron, and what was the function of chylomicron? To move the lipids from intestine to the periphery, including lipid and muscles, and from there on it takes it to the liver. So it takes it to so from intestine it moves it all the way to periphery, which includes lipids and muscles, for instance, and then from here on it takes it to liver. The function of VLDL and LDL, on the other hand, was to move lipids from liver to periphery, and that's bad. We don't really like this, so let me write this as bad. And then finally we have HDL, which moves the lipids and cholesterol all the way from periphery to the liver, inside which they could broken down or get excreted as bile salts inside the intestinal tract. And this is actually very good because it gets rid of excess cholesterol that can cause blockage of the vessels. Alright, so basically that's about it. That concludes our discussion. We have discussed the functions and structures of the digestive system. We have done some practice questions. Of course, if you need more practice, you can always refer to our question bank. I'd like to thank you all for listening and see you all for the next video.